Hey Team Bio, welcome to your Chloroplast structure introductory video. So when we come back from break, we're going to dive into the study of photosynthesis and cellular respiration. Let's really quickly write the unbalanced chemical equation for photosynthesis. Photosynthesis, our inputs are CO2, which is a gas in the air, plus H2O, which is a liquid that's coming from the ground through the roots of plants, plus energy. This energy is electromagnetic energy from the sun, and we get out C6H12O6, which is glucose and oxygen. This is unbalanced. You can balance it if you want. Now, cellular respiration. And those of you not on iPads, don't hate me too much. We take the products of photosynthesis and they become the reactants in cellular respiration. And then we take the reactants of photosynthesis and they become the products in cellular respiration, except we change this energy form from sun to ATP. Okay, so we're going to dive into these two reactions. We're going to figure out what's happening at every step and how we go from these inorganic molecules, CO2, H2O, and energy from the sun, and we become organic, and then how do we break down this organic molecule back into CO2 and H2O. So we're really going to just get into the weeds here. But first, we need to understand the structure of the chloroplast, because the chloroplast is where photosynthesis is happening. So that's what this video is all about. Okay, so let's get right down to it. So the chloroplast is actually a triple membrane system. It's composed of 3 lipid bilayers. Incredible. So the outermost lipid bilayer is shockingly called the outer membrane. So you all know the structure of the lipid bilayer by now. If we were to zoom way in on this outer membrane, we'd see some phosphate heads here on the outside, and then we'd see some phosphate heads also on the inside, and then there would be some tails attached to those phosphate heads on both sides. So it's a lipid bilayer, the outer membrane. Okay, the next lipid bilayer that's just interior to the outer membrane is another membrane called the inner membrane. Just like the outer membrane, it's composed of phosphate heads, tails, heads. What we're doing here is creating a series of compartments that are going to be functionally different from one another. So the spaces inside of these membranes are going to be areas where we're going to actually build up concentration gradients and use those concentration gradients to power stuff in the system. Okay. Inside the inner membrane is a jelly-like liquid that you can it's like analogous to the cytoplasm of the chloroplast itself. And it's called the stroma, S-T-R-O-M-A. Now, this is very confusing, not yet, but there's a structure in the leaf that is called the stomata or stoma. Singular. And so you're going to confuse, some of you are going to confuse those two. Don't confuse the stroma, which is the similar substance like the cytoplasm of the chloroplast to the stomata, which are little holes in the underside of the leaf. So stroma, jelly-like interior of the chloroplast. Okay. And so in the interior to the innermost membrane is our last membrane, and it is right here. I have it in dark green, and this is called the TMS, which is an abbreviation for thylakoid membrane system. Okay, so this is the thylakoid membrane. The whole thing, like the interior, I guess, as well, will be part of the thylakoid membrane system. Or I guess you could just say thylakoid membrane system is going to be what's in the membrane and then also what's inside of this structure. So we'll put my little thing like that. So you'll notice that this is a highly folded membrane, and the reason for that is we're trying to maximize surface area to volume ratio, get as much surface area as possible on this membrane because it's like the action membrane of the chloroplast. It's going to be where all the reactions of photosynthesis are taking place. Okay. You'll also notice that there are kind of like, I haven't done the best job of drawing it, but little kind of jelly beans that are all connected on the interior. Your textbook does a better job of actually making them look like jelly beans. So they're all interconnected, but they look like little blobby discs. One tiny little disc So this is one disc here. This is called a thylakoid. And a stack of discs, all one on top of another, from here down to the bottom here. This is called a granum. I don't know why. It sounds like a granny to me. Okay, so we've got the thylakoid. Oh, shoot. We've got the granum, which is the stack of thylakoids. And the space that is in the very interior of this membrane system, the TMS, is called the lumen. You're going to see this word lumen when we talk about other structures as well. So like the interior of the rough endoplasmic reticulum is called the lumen, for example. Okay, so only one more thing to label, I guess, in the chloroplast is that the granum, they're all connected one to another through these kind of little channel-like areas that are called lamella. So here's a thylakoid, a stack of thylakoids makes a granum, and then granums are connected one to another through these structures called lamella. And these all make up the thylakoid membrane system. So maybe I should have indicated that somehow, but the TMS is composed of all of these things. Can we put a big curly braces around them? Not very easily. Okay, so I think that is it. So your next screencast, we're kind of going to do is zoom out, and we're going to put the whole chloroplast in context. So we're going to say, okay, here's my zoom out box. Whoop. So we're going to look at the cross section of a leaf. And see where the chloroplast is within that cross section and see how the leaf is structured so it can maximize photosynthesis. The leaf is actually not a single layer of cells. I know it looks really thin and tiny, but it's actually multiple layers stacked on top of one another. And then we're also going to do a zoom in on the TMS itself in the screencast that follows the cross section of a leaf screencast. So we're going to see what's happening within the TMS, and that's going to help us answer the question, like, what's happening, what are the reactions that are powering photosynthesis, and how are they working with the structures that are there. Cross-section of the leaf, zoom out, and structure of TMS. Zoom in. And this is a zoom out. I don't know why I'm writing it in pink. Okay, that's all. See you in class next on Wednesday.